Hi, my name is Maia Machiela, and I'm here on behalf of Dr. Adam Locke talking about the effect of feeding rumen-protected methionine and lysine with or without an animal-derived source of rumen undegradable protein on the performance of mid-lactation dairy cows. So just to give us a big picture overview, in most markets, milk income is driven by the yields of milk fat and milk protein. Therefore, if we can understand how different amino acid supplementation strategies affect production, we can make feeding decisions based on this and help to improve the efficiency of the dairy cow. So why should we balance amino acids? Well, in typical dairy cow diets, methionine and lysine are the most limiting amino acids. By supplementing methionine and lysine, we can meet the requirements for milk protein synthesis without oversupplying crude protein. This can potentially improve nitrogen efficiency and decrease nitrogen excretion. Rumen-protected methionine and lysine are often fed to help increase amino acid availability for absorption and mammary gland utilization. We generally see that methionine and lysine supplementation increases the yields of milk and milk protein, and we also generally see a reduced efficiency as the level of supplementation increases. This is because excess amino acids are excreted through ureagenesis, which is an energetically demanding process. Different production responses have been observed, though, due to differing dietary crude protein levels and energy availability. So the regulation of milk protein synthesis is complex. Amino acids like methionine and lysine generate a response because, at some point, they are the limiting amino acids for protein synthesis in the mammary gland. However, there are other amino acids, such as leucine and isoleucine, that can change the metabolism of the mammary gland and accelerate milk protein synthesis, thus having a stimulatory effect that improves efficiency. There are also other dietary factors, such as starch or fermentable carbohydrates, that can change milk protein synthesis, but we will not be focusing on those in this study. So moving beyond just the supplementation of methionine and lysine, Yoder et al. Infused methionine, lysine, and histidine, shown in orange, leucine and isoleucine, shown in gray, or all 5 of those essential amino acids together, shown in yellow. They observed an additive response on energy-corrected milk, as you can see here, when they supplied all 5 of these essential amino acids compared to only methionine, lysine, and histidine, or leucine and isoleucine. They concluded that methionine and lysine are important amino acids, but the supply of other essential amino acids can improve their utilization. And this led to the basis for our study. If we provide more total essential amino acids in the form of rumen undegradable protein associated with methionine and lysine, we could also potentially see that same additive response that Yoder did. To date, we do not know any studies comparing these two methods. So therefore, the objective of our study was to determine the effects of supplementing rumen-protected methionine and lysine with or without an animal-derived source of rumen undegradable protein on the performance of mid-lactation dairy cows. We hypothesized that supplementing rumen-protected methionine and lysine will increase the yields of milk and milk protein, and that increase will be greater when additional essential amino acids are supplied through the use of rumen undegradable protein. So moving into our materials and methods. We used 36 cows that were assigned to a 3x3 truncated Latin square design. Our treatments consisted of 3 diets, with the first being a control diet with no supplemental amino acids. It will be shown in blue text throughout this presentation. Our second diet was a diet supplemented with rumen-protected methionine and lysine, ML, shown in green, and our last diet was a diet supplemented with both rumen-protected methionine and lysine and an animal-derived source of rumen undegradable protein, MLO, shown in orange. Our experiment consisted of 2 35-day periods with a 5-day sampling period at the end of each period. Just to give you a visual of our data collection throughout the study, from days 1 to day 31, we measured body weight 3 times per week. And from day 31 to day 35 was our sampling period, where we collected milk, feed ingredients, and orts daily. We took 2 blood samples on the last day of the period, day 35, along with body condition score in those cows. Body weight continued to be measured 3 times per week. So just to dive a little bit more into my experimental design. So in a traditional Latin square, if there are 3 treatments, there should be 3 periods, with all cows receiving all 3 of the treatments, as you can see in the table here. However, in these mid-lactation cows, we wanted to have longer periods to observe the treatment effects on body weight and long-term production. Those longer periods, at 35 days each, would cause more variation between treatments and weaken our stats. Therefore, to overcome this, we followed a truncated Latin square design with 3 treatments across 2 periods. By having sufficient squares, or blocks, this balanced for treatment sequences in periods 1 and 2. This also gave us double the statistical power compared to a block design. Moving into the composition of my diets. In terms of feed ingredients, we formulated all 3 diets to have the same amounts of corn silage, alfalfa silage, wheat straw, high moisture corn, ground corn, soybean meal, cottonseed, urea, a palmitic acid supplement, and a vitamin and mineral mix. Where our diets differed was in the use of dried distillers grains, soybean hulls, and for the second diet, we had a rumen-protected methionine and lysine supplement. And for our third diet, we had a rumen-protected methionine and lysine and essential amino acid supplement. Both amino acid supplemented diets were formulated to provide 24 grams of lysine and 14 grams of methionine, respectively. In terms of nutrient composition, again, all 3 diets were formulated to contain the same amounts of forage NDF, starch, estimated energy, and crude protein. Where our diets differed was in RDP and RUP, with diets 1 and 2 having 10% RDP and 6.4% RUP, and diet 3 having 9.7% RDP and 6.7% RUP. Our statistical model included the overall mean, the random effective cow, the fixed effective period, the fixed effective treatment, the fixed effect of the interaction between period and treatment, and the residual error. Main effects were deemed significant when p-values were less than or equal to 0.05, and tendencies were defined when p-values were less than or equal to 0.1. Our statistical model also included 2 pre-planned contrasts. The first being control versus protein treatments, which looked at the overall effect of amino acid supplementation. Our second contrast looked at the effects of the individual treatments, ML versus MLO. Now turning to our results. So to quickly orient you, my treatments will always be on the x-axis, my dependent variables will always be on the y-axis, and my p-values will always be directly underneath each graph. For dry matter intake, we saw no statistically significant differences. In terms of milk, I will first talk about milk yield, and then I will move into the yields of milk components, and finally I will talk about fat-corrected and energy-corrected milk. Overall, we saw that protein treatments tended to increase the yields of milk by 1.2 kilograms per day. We saw no statistically significant differences between ML and MLO. Moving into milk protein concentration and yield. We saw that protein treatments increased milk protein concentration by 0.08%. We also saw that protein treatments increased the yields of milk protein by 0.06 kilograms per day. We saw no statistically significant differences between ML and MLO. For milk fat concentration and yield, we saw that MLO increased milk fat concentration by 0.18% compared to ML. For fat yield, we saw that protein treatments increased the yields of milk fat by 0.05 kilograms per day. And we also observed that MLO had a tendency to increase the yields of milk fat by 0.04 kilograms per day compared to ML. Moving into fat-corrected and energy-corrected milk, we saw that protein treatments increased the yields of fat-corrected milk by 1.4 kilograms per day. We also observed that protein treatments increased the yields of energy-corrected milk by 1.4 kilograms per day as well, compared to control. We saw no statistically significant differences between ML and MLO. For body weight change and body condition score change, we saw no statistically significant differences. An interesting piece of information that we observed was our milk fatty acid yields by source. So interesting enough, there is some research indicating that amino acids can stimulate de novo milk fat synthesis. And when we fed more total essential amino acids in our diets, especially diet 3, we provided some of those amino acids that are needed. So for de novo milk fatty acids, we saw that protein treatments increased the yields by 24 grams per day. We also saw that MLO tended to increase the yields of de novo fatty acids by 13 grams per day compared to ML. For mixed milk fatty acids, we saw that protein treatments increased the yields by 34 grams per day. And we also saw that MLO increased the yields by 23 grams per day compared to ML. Lastly, MLO decreased preformed milk fatty acid yields by 18 grams per day. These results deserve more investigation. So to conclude, we saw that both amino acid supplemented diets, ML and MLO, increased the yields of milk fat and milk protein, but no differences were observed between the two diets. Compared with ML, MLO increased the yields of mixed milk fatty acids, tended to increase the yields of de novo milk fatty acids and milk fat yield, and decreased the yields of preformed fatty acids. So a quick discussion on why we may not have seen any differences between the two amino acid supplemented diets. So one thought is that the difference in supplementation between the two diets was not enough to observe a significant difference. Another thought was that crude protein was formulated at 16.5%, giving us a possibility that this provided close to adequate or adequate levels of amino acids required for milk protein production. The effects we observed of amino acid supplementation on the yields of milk fat and fatty acid sources deserves further attention. I would like to thank the Locke Lab Group for all of their work on this trial and the MSU dairy farm workers for their day-to-day care of these cows. Lastly, I would like to thank Purdue Agribusiness for their financial support of this trial. If you have any questions, please feel free to email me at the email listed. Thank you so much for listening.